So when you say that enzyme is called biological catalyst. It's something like catalyst, but it's a biological catalyst involve the proteins. Okay, so enzyme increase the speed of chemical reaction without undergoing any net chemical change. Highlight the word net chemical change. So means that during the process, they may have the chemical changes, but at the end of the process, they won't change. Are you clear? They won't change the chemical structure at the end of the process. And generally, all enzymes are globular proteins. Be careful, all enzymes are protein, but not all proteins are enzymes. Okay, be careful. Enzyme is a subset of protein. So it means that if we learn about the globular protein, we learn about this. When globular proteins, they must be water soluble, and makes sense, right? Because chemical reaction take place in the aqueous medium. If let's say our reactant dissolve in water, but enzyme cannot dissolve, then it doesn't make sense. Okay, so how the structure take place? So structure actually maintains by the tertiary structure and the quaternary structure. Very important. The spherical structure maintained by the protein tertiary. Quaternary basically means that some enzyme actually require more than one polypeptide, so they become a quaternary protein. Okay, so this spherical structure is induced by the protein tertiary and quaternary structure. Hydrophobic amino acid will bound towards the molecule interior, and the polar amino acid bound outwards, facing the aqueous medium, allowing the formation of hydrogen bonds with water molecule, making the enzyme now soluble. Okay. Now, this is something extra for you guys. Even though most of the enzymes are protein, but we do have some type of RNA molecule have the catalytic activity as well, right? RNA molecule, but we don't call them as enzyme. Are you clear? Right? We don't call them really enzymes because they are RNA. So enzyme names always end with ASE, unless before we use this system, we already named them. For example, pepsin, lysine, autolysine, expansin. Those enzymes, we already give them the name before we start using the ASE. Okay, so nowadays, how you determine whether they are enzyme or not? So they end with ASE, they are enzymes. Okay, the proteins molecule, we give the name, and if ASE means that they are enzyme. So in living organism, enzyme can classify into two broad categories, either intracellular enzyme or extracellular enzyme. So what is the difference, or what are the differences between these two enzymes? Actually, is quite clear. Okay, depend on location where the enzyme functions. So for example, intracellular enzyme will work inside or catalyze the metabolic reaction within the cells, but extracellular enzyme catalyze the reactions outside the cell. So means the enzyme must be secreted out from our cells. Okay, so example intracellular enzyme, for example, protein kinase A. Okay, some enzyme that you're going to learn. For example, what is this enzyme? Carbonic anhydrase. Now you may ask me, do I need to remember? The answer means you're going to memorize. Why? Because over the times you will be able to know the enzyme name already, because we keep on using that name. Carbonic anhydrase, protein kinase A, those enzymes you automatically will register in your brain. Okay. Now, how about extracellular enzyme? Your amylase, pancreatic amylase, for example. What else? Your trypsin, pepsin, okay, sucrase, maltase, okay, lipase. Those are, you need to secrete out, right? So those are the extracellular enzymes. Okay, so with this, I have done for the general part about the enzyme. So I off the record now.